day. This is the Professor, CBMD PhD, and today is Wednesday, August 23rd, 2023. It is 2:36 and 40 seconds, a.m. Eastern Standard Time, and today's research, brain tumors. So let's hop to it here. Improved survival for incurable brain tumor, providing a crack in the armor. Date August 16, 2023. Source: University of Michigan. The scientific investigators and microbiologists have found a potential drug candidate that improve outcomes for patients with a type of childhood brain tumor for which there are no effective treatments. The compound called ONC-201 nearly doubles survival for patients with diffuse midline glioma or diffuse intrinsic pontine glioma compared to previous patients. The diffuse midline gliomas, including diffuse intrinsic pontine glioma with a mutation called H3K27M, are particularly aggressive, with an overall survival rate of 11 to 15 months. These tumors are most frequently found in children and young adults. The only available treatment is radiation, and even that is difficult, as the tumors are located amid brain regions with critical functions. It is incredibly difficult tumor to treat. Prior to this study, there have been more than 250 clinical trials that have not been able to improve outcomes. This is a major crack in the armor. In 2 clinical trials testing ONC-201 in a total of 71 patients with H3K27M mutated diffuse midline gliomas, the median overall survival was nearly 22 months for tumors that had not recurred at the time of enrollment. Almost a third of the patients lived longer than 2 years. ONC-201 took an unusual path to a clinical trial, initially designed to target dopamine receptors, which are unregulated in many different tumors. The researchers saw that the drug passes the blood-brain barrier, one of the biggest challenges to designing drugs for brain tumors. Additional trials in glioblastoma were not successful, but a small number of patients with diffuse midline gliomas that carried the H3K27M mutation had more promising results. Without understanding why it worked better in these patients, a phase 1 trial was started in children and young adults with H3K27M mutated diffuse midline gliomas. And through the trial they collected cerebral spinal fluid from patients. They used this fluid to analyze metabolic changes and found ONC-201 got into the tumor cells and affected mitochondria. Patients who responded to the drug had an increase in a metabolite called L2HG produced by tumor cells. The researchers called the finding very much unexpected. The team found that increased L2HG reversed tumor-defining epigenetic signals, causing tumor cells to differentiate more and divide less. The longer patients were on ONC-201, the more tumors exhibited these epigenetic reversals. And this could explain why this patient population was responding so well to the drug, because it had a specific epigenetic abnormality that could be turned off by ONC-201. The tumors have an epigenetic change caused by H3K27M mutation, and ONC-201 metabolically undoes that change. The researchers know that even a near doubling of survival is not enough for families of patients with this diagnosis, as the tumor remains very lethal, but they hope this first step will lead to bigger leaps in the future. As for now, we have this patient population that didn't have a drug before, and now we see many of the tumors responding. They have a platform to build on, and they can also explain why it is working. They are really excited about this study and envision ONC-201 becoming standard of care for these patients in the near future. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Leader of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.